Today is all about annotation and data analysis, especially if you have information-rich / noisy images. So Matt Swulius will take it over. He's coming from Penn State. And what is your favorite part of the cryo-ET workflow?
I love data collection. I like seeing things that I've never seen before. And so just anytime something new goes into the microscope, I get excited to go see what's happening.
You're a visualist,
For sure.
Take it away.
All right, thanks. Well, hello, everyone. I know we haven't all officially met, but I'm Matt Swulius. To talk today about using both synthetic and real ET data for using train deep learning models really for segmentation, because that's where we got stuck really. I mean, collecting data is not a really hard thing for us. Been doing it for a long time. We just pile up lots of tomograms and we don't know what to do with them. And then I'm the director of the core facility at Penn State College of Medicine. And so I collaborate with like 5, 6 other labs that are all doing different things, looking at different systems, in vitro, in vivo, whatever. And what I realized quickly was that I could get the data for them, but I couldn't make sense of it all for them quickly. And they weren't going to do it on their own. And they were all relying on me to analyze this data. And so when I realized what my role was, we started turning towards, building, segmenting tomograms and trying to build cellular models. I try to think of it now like, fitting an atomic model into a density map. We're trying to build the best models of the tomograms we can so that we can come back to them with the computer and ask questions about what's going on in that scene. And so we're lazy, or I'm lazy. That's a good thing because nobody wants to actually segment hundreds of tomograms. And so Carson in the back over there coded this software for us. We call it CryoTomo Sim. It's a MATLAB-based software package that can take in any PDB that you want, or any atomic model really, as a PDB or a .zip. So it doesn't have to come from the PDB. You can go to AlphaFold. You can do whatever you want. You can assemble your own models in Chimera out of whatever you want and feed them into this. But here we have a proteasome, ribosome, and this actin filament. And you basically just put in what pixel size you want, how large of a volume you want, whether you want vitreous ice turned on or off, membranes, all these different sort of modeling features. And it spits out a synthetic tomogram with all of those objects in it randomly placed, and you can control the density of them and the numbers of each of them and things like that. So you can kind of create your own recipe to make a synthetic tomogram. This is a projection through one of the models that CTS generated. I just like to show it because it kind of shows off the things that it does. I think I have them labeled here. All right, so macromolecular assemblies. This is basically anything you can imagine. In this case, what we have is a microtubule segment. And inside of it, you can see there's one little intraluminal particle. I don't know if you're familiar with MIPS or microtubule intraluminal particles, but they're in neurons a lot. And so there's 2 here, there's 2 here in different stages, there's 3 in this one right here. And so we built a model that has 3 MIPS inside of it. We tagged it as an assembly for the software, and when it places each of these, it randomly places MIPS inside of it. And so you can build whatever you want in, say, Chimera, and define different sub-models and send them in, and it will sort of put different instances in a sense. It'll put in the initial model, like whatever the first submodel is, like in this case, it was a microtubule. Sorry. And the subsequent models will be randomly selected. Linear bundles. So it's kind of hard. This is a very crowded model, but you can see that these actin filaments, say here, are actually running in a bundle together. A lot of them are. And so you can turn these bundling features on and off for these filaments. You can embed membrane complexes into these spherical vesicles that we make. So these are, you can turn these vesicles on and off, so you can have them or not. It's hard to see, but there's actually transmembrane proteins embedded all through them. You can see some here. Now, it's going to be a little bit laggy, but this is actually a tilt series. So a tilt series of projections through this model that CTS generated. We then corrupt this tilt series with a variety of things. So one of them is a defocus gradient. So as this thing tilts, you know, in tomography, you don't have a constant defocus across. You have this sort of gradient. And so you can see this is under focus and this is over focus. So this is just a high tilt. We can turn on the detector and actually get this movie is, yeah. This movie stalls sometimes, but you can get them. These are noisier than they were before. And you end up with this tomogram that you can now scroll back and forth through. Now, the really fun thing about it is that you also get this map of where everything is. And this is where I said we're lazy. Like, we don't want to have to annotate every one of these objects. So in our simulations, we know where they all are. And you can sort of see how they line up. Now, let me see what's here. So we use these for segmentation, right? So we now have this synthetic data set and we have a ground truth for it. And so we need to train something. I'm going to talk today about everything in Dragonfly. I do not work for Dragonfly. I just use it a lot. And everything I do is going to be in there. But this is not set to be done in Dragonfly. You can take all of these outputs from CTS, it's cryotomosyn, that's what I'm going to call it. You take all these CTS outputs and put them into whatever deep learning pipeline you want. But for all of these models, we were using patch size of 128 by 128 by 11. Batch size of 8. We'll talk about this later today, so you don't need to really get all of this. We'll actually go through setting this up, and you have these slides, but these are just the parameters that we used. Now, we have a simulator, and we can do things that you can't easily do without a simulator. And so we decided we should try those things first, which was like, we wanted to test how different optical parameters in the microscope affected deep learning, right? So this is just a series of ribosomal tomograms. They're the exact same model, but they're projected at different doses of electrons. And so it's like low signal to high signal. And we trained the network to segment at the high signal, and then we inferred it to the rest of them. And you can see it falls off pretty quickly, and you can see it in this dice score graph, the easiest, right? Like it starts out up high on the right and it falls off. Now, if you do this opposite and you train on the lowest signal data and then you infer back the other direction, you get this flat line. It's like it works pretty well under all conditions, but it never gets as good as this other network. And you can see that in the way that it segments these ribosomes if you zoom in on them. Like, it just depends on how much of a completist you are in a sense. These are just like really nice silhouettes of like the ribosomes, where these are kind of, anyways. So why not just train on both? That's what we do now. We train on both, and you get this network that kind of does well in all cases. And that's true of defocus, even though defocus is not nearly as prominent, right? So actually, all of these look like they were pretty well segmented if you look at the blue. And they were like these were trained at really low defocus and then went to high. This was trained in the middle somewhere, and you see it kind of fall off in both directions. But together, you get this kind of curve here. Anyways, and the same thing is true for pixel size. And so I won't belabor the point, but obviously, you know, training across pixel sizes to some degree is helpful. And so what does this look like? So this is inside a dragonfly. This is a synthetic tomogram with cophylactin and actin filaments. Okay, so actin is, I'm sure most of you have heard of actin. Cophylactin is just actin decorated with cofilament. Someone came up with a nice name for it. Oh, there we go. So this is what the ground truth looks like when you lay it over it, right? So this is the CPS outputs in Dragonfly. And you can see that the red is actin and the cophylactin is orange, but it has these cofilin molecules decorating the outside of them. Now, if you train a network to find them, it initially finds them all as actin, and then it starts struggling, goes back and forth. But eventually it figures out how to segment all of those classes. And if you then take a real tomogram, this is just in vitro polymerized actin, then mixed with cofilin, you actually get this red, like the bare actin filaments get segmented as bare actin, and cophylactin gets picked up, and it actually gets these spiral cofilins around the outside. I mean, I don't know that it's literally finding cofilin, but it knows where they are. It just kind of makes me happy. And there it is in 3D. You can see, obviously, it gets some noise in the background as well, but and just to show that it works on a variety of things, this is a segmentation of actin bundled by this protein called CAM kinase 2. So that's what these little blue specks are. Makes this huge bundle. And all I did was put actin and CAM kinase 2 into CTS, and it spit out this synthetic tomogram and a ground truth, trained the network, and it was able to pick out all of that. And if you look closely, it even gets like the helical twist in the actin filaments if you look at like single actin filaments. I mean, it's certainly better than I can do by hand, is all I'm trying to point out. You have membrane-bound stuff. This is ATP synthase over here. So this is segmentation of a real tomogram of, what do they call them? They're like mitochondrial vesicles, right? So these are basically the inside cristae sort of stripped out of a mitochondria and then plunge frozen. And so I just took our vesicles and jammed a bunch of ATP synthase into them and trained a network to find both the membrane and the synthases. And it does a surprisingly good job. I mean, I wouldn't do that by hand. I would never even try to do it, to be honest. But we run into issues when we get into cells, all right? So this is a cellular tomogram, obviously. There's ribosomes and a bunch of microtubules running north-south in there. And if you just train a network to find microtubules, that's what it does. I mean, it finds everything. It looks pretty crazy, but it does find the microtubules. And so we reasoned that it's probably just how crowded this environment. You know, there's a bunch of things it hasn't seen yet, so it's just picking them. And so we took out a chunk of that. And that's what's over here. We hand segmented it to generate a ground truth for these pieces of microtubule. And we trained 5 different networks. And we segmented that section with these different networks. And you can see the emergence of the microtubules and the sort of washing away of the background. Now, and this is all done with simulated data. We didn't have to hand segment anything other than our ground truth. But the initial one is what we started out with. It has no background to it, no distractors. It's just microtubules. Then we started adding little small proteins to it. So these are just little less than 200 kilodalton blobs. And you start to see things emerge. These are three-dimensional renderings of the segmentation, just so you know. But anyways, you add more protein, you add macromolecules, it really cleans it up. And when you put them together, it's as good as it can get without, introducing real data. And so we tried multi-class networks. This is a single network trained to find 4 different classes: ribosome, microtubules, actin, and membranes. These are 4 different tomograms. These are 2 neuronal tomograms. This is a fibnil HeLa cell and some C. elegans embryo that I just took off the internet. But what you can see is like this single network actually does a surprisingly good job. And you never had to do any real work. Now, it's not perfect by any stretch. Like, for instance, you see a lot of these microtubules bending over the carbon get picked up as actin, so the blue turns to green. Or this mitochondrial membrane here, like it's never seen that, it doesn't know what it is, so it finds it as actin. And if you go through there, there's plenty of those little mismatches. You can find them, but it's very clean. And so we wondered, could we clean it up by adding some real data to it? But we don't want to do a lot of work. And so we tried doing minimal work. We took a little piece of a tomogram. This is a segmentation of a real tomogram. And we have same thing, microtubules, membranes, ribosomes, and actin. We then trained 4 different networks. One of them that was trained simply on a single slice of the ground truth. And that network failed spectacularly. Now, anyway, we could get into reasons for why it did, but under these conditions, it failed. It didn't segment anything. So it all had a dice score of 0. That's why it's. But if we took 5 simulated slices of what we made simulated cytoplasm, right? So what we did is we packed a bunch of stuff into a synthetic tomogram and trained it to find these 4 things. And it does an okay job. It's not nice. That's just, and then if we add 10 times more simulated data, it actually gets significantly better. And then if we add just that single slice that failed to train the first network and we dual train, you see that these dice scores all go up. And so just peppering in a little bit of real data does help significantly. But the nice thing is that if you have segmentations like this before you start out to train your networks, you're already in pretty good shape, because all you have to do is go in and hand correct some of the mistakes that the current network is making and feed that back into your training. And that's what we'll go over this afternoon. So we trained using this sort of co-training approach, we just segmented, it was 106 tomograms, right? So the 106 tomograms from neuronal growth cones. This is just an overview of one of these neuronal growth cones. This is actually like the end of a growing axon. And anyways, there's 26 different tomograms collected here. They were all segmented by a very small number of networks. And we were able to compile some information about the distributions of actin and coprolactin and ribosomes and TRIC and various things throughout the growth cone. Now, that was cool, but we wanted to make one network that could kind of do everything, or at least we wanted to try. And so we moved on to trying to train a very generalized network. And so for this, we actually have a block of data. Let me see. Yeah. We took 10 simulated tomograms. They're all just mixtures of 7 different classes, right? So membranes, TRIC, ribosomes, actin, coprolactin, microtubule. And the background always gets but we simulated these tomograms of 10 different models with random defocus values. I mean, within a certain range, they're not wild, but within a reasonable range, we have randomized all of the optical parameters so that these 10 tomograms have a large variation in their content and their simulation parameters. And then we concatenated them just to feed them into Dragonfly as one block of data. It's simpler that way. And there's other reasons we do it later, but okay. So in our first iteration of trying to train this sort of more generalized network, we took our tomograms of neurons and we ran our initial network on it, and it does okay. It's not getting all of the membranes. I take that back. Ling made these slides, and so I forgot what they were. Okay, we start over. On our first iteration of this generalized network, we had this already segmented chunk of this real tomogram. And so we took it and we added it to our synthetic data. And we trained an initial network. And it was okay, but not perfect. This is that same sort of crazy tomogram with all of the microtubules running north-south. You can see that it's trying for sure. If you zoom in, it's definitely mismatching some actin and microtubules and things like that. And so we would hand correct it. And I'll show you how to do this in Dragonfly, because you can just go in and say, I want you to change this class to this class, right? Same thing on another tomogram. You could see that this is a bundle of coprolactin running through the tomogram. It's misclassifying some of it as actin green. And so we would hand correct these kind of differences and we'd feed it back into the network. And we'd sort of repeat this, right? We had like a set of tomograms. We would then go into non-neuronal tomograms. So we had like these eukaryotic multi-layer membranes. We have a bacterial double membrane here. And then this is just some other eukaryotic cytoplasm where we had to strip certain misclassifications out and train the network to ignore those things. And then on top of that, what I haven't shown you is that it finds all kinds of junk, right? You know, like the top of your ice, the carbon surface, the edge of the carbon. So what we did is we had a final iteration where we went in and picked those sorts of things out of our tomograms, like ice on the surface. I said this already. And we just collapsed all of that into a background class, and we fed that back into our final network, which has this very gray sort of block in here where it's all background. And anyway, what came out of it, we got this network we call neural seg. Neural seg can segment, I don't want to say anything, okay? But it can segment a lot of things. And certainly to the degree that you might want to put it in for visualization, right? I mean, it can segment a lot of things for visualization. But so these are microbes. These are actually different types of bacteria where it's finding ribosomes in the double membrane. This is a archaeal cell where it actually has some archaeal actin filaments in it. You can't really see them, but they're in there and it's getting picked up by the network. This is some is a blue-green algae, retinal pigmented epithelial cell, intact neuron, and then this is actually brain tissue, sort of milled recently out of the Goal lab. And, you know, like I said, it can't segment, well, it can differentiate between actin and coprolactin, though. So you can see, like in this tomogram of actin and coprolactin, this is an intracellular tomogram. Let me go back one. Kind of hard to see on this screen, but like there's a bunch of bare actin filaments with these bundles of coprolactin. The coprolactin are these squiggly actin filaments. It does quite a good job in situ, I should say, sort of delineating coprolactin from actin and pulling out bundles of coprolactin, individual filaments intercalated within bundles. It's a pretty robust model. And like I said, it ignores all kinds of things, like the carbon edge. So here's this crazy carbon edge. It always gets identified as a microtubule or some like membrane. And with the carbon distractors, it disappears. Look at all these gold fiducials. I mean, that should be data wasted almost. I mean, not really, but it can just see right through them and pull out all of the microtubules in there. This is not the most beautiful figure, but these are 4 tomograms collected in similar regions at different defocus values on our microscope. The same network running across all. Now, it does a pretty good job at all of them, but this is the same tomogram resampled at 6 different pixel sizes, from 10 angstrom per pixel all the way to 1, run through the same network. Now, it's definitely not perfect in all cases either, but certainly for visualization, you're doing all right. For quantification, like if you really look at this, you can see that a lot of the small molecules, like the ribosomes and the trig chaperones, kind of fall out as you get too far. We trained this network between 12 and 14. So the take-home message is it does the best at 12 and 14. But it does okay in all of them. Electron dose. So it works fine around 150 to 200 electrons total. That's what we often collect for cellular tomograms, but we just cut 1 in half to 90, and it still works pretty well. 45 was, it depends on thickness of your tomogram too, right? It's like a balance of those things. All right. I'm going to talk a little bit about, yes.
I just had a quick question.
Yes.
Do you think that the neural network would be able to distinguish between like filamentous actin and monomeric actin?
So I have never trained it specifically to try to do that.
Yes.
But yes. Well, I was going to make it too complicated. Carson, it gets right to the.
Yes.
Okay.
Yes.
Okay.
You have a good enough image, you can put segmentation. Fine.
Okay, thanks.
Yeah, sorry. Thank you, Carson.
I don't know.
What I would do, I mean, in that case, I would probably train a network specifically to do that. I wouldn't, this network will not.
Okay.
This network was not trained, this neural seg was not trained to find filamentous or globular actin.
Mhm.
But if you had tomograms where it was visible, we could train a network that would.
Okay.
Now, how easy is it to distinguish actin monomers in a cell from some other thing? I think that's where it gets really tricky. But in an in vitro situation where you wanted to maybe get some sense of how many monomers were floating around in your buffer, actually, I think it can do it. And we'll see that maybe this afternoon. Any other questions? I should have said, feel free to, this is a workshop, so feel free to chime in anytime. All right, there's been talk about denoising. I'm going to just talk about what we're doing. And it's based on, Alistair made a very good argument yesterday for the fact that we do not have perfect cryo-EM images to train a neural network to predict, right? Like, you have a noisy image, but you do not have a perfect version of that image that you could train the network to predict. But we built this simulator. Kind of a crazy idea, but we were like, we can actually do things that you can't do with a real microscope, with real cryo data. And so here's a simulated tomogram of ribosomes. And here is the ground truth, or what we call the ground truth.
What kind of noise do you add? Is it just like Gaussian or?
We don't add any noise specifically, all the noise is arising from EMG. So like the CPF, the ice having variable density and thickness. the random distribution of electrons being sampled, that sort of stuff.
Okay, and so the simulated ones, you set those other parameters, you think, so, like, okay.
Yeah, there isn't a, I mean, I guess the closest thing to a noise parameter is the dose.
Okay.
You just turn that up and down.
Like between those and.
So you can control all of that. In this case, what I call a ground truth is a tomogram that doesn't have a missing wedge. And it has very, what is this term? I call it infinite dose, because I always do just like breaking them out. I like it's a quote unquote perfect projection. And there is some tomographic noise still in here, just from the reconstruction. Obviously, there's like a little, you can see some noise in here, so it's not perfect. I have tried denoising to just the output model from CTS, and it's pretty crazy. It doesn't really work well. But in this case, right, I can train a network to regress from that noisy tomogram to its ground truth. Now, here's a new tomogram of ribosome. This is synthetic again. This is our network's regression of it back to our ground truth. Now, it's obviously denoises like in a very harsh way. Like it literally strips the noise out of it. But we were interested in whether or not we could apply this in some general way to cellular tomograms. And so we started by making synthetic cytoplasm. So we would start with membranes. I'd add a layer of cytoskeletal elements. And I do this because these are big. And as you start crowding molecules into the model, as you're trying to put new things into it, these things get really hard, because if it clashes even with a tiny actin monomer that's in your model, it'll just like reject that. And so we start with a layer of cytoskeleton, actin, and microtubules here. Then I'll drop in a bunch of like large macromolecules. And usually I've tested to see like how many should I have not like rigorously tested how many do I need to add to get the best network. What I know now by my like just playing around with things is that if I had 20 different macromolecules from the PDB, I don't know how much it helps me to add like 100. What do you think?
That's understandable, but generally not much.
Yeah, like by my eye, I just don't see that much of a difference. And so what I have here is like 20 different macromolecules. And then I dump in a bunch of little ones. Anyways, we then generate synthetic tomograms from those models, and we calculate ones with different defocus values, different signal to noise. This is where it's up to you to decide like how crazy you want to be, but you could really search a lot of parameter space, pixel sizes, and all kinds of things and try to make a really generalized network. But all of the network, so this will be like the input to the network, and it's all being trained to this ground truth. And this isn't going to play really well, but here's a tomogram at the leading edge of the neuron. We'll play around with this tomogram later. It gets used a lot, but you can see the tubules and everything, but it's noisy, and we run it through our denoising algorithm, and it gives you something like that, which is, it hurts your eyes at first. It's a little weird to look at, and I feel the screens are chopped with that. For things like particle picking and just like finding things that you need to find in your tomogram, it actually even preserves pretty good high resolution information. These are coprolactin filaments running through right here, and this is a bare actin filament. I don't know if you can see the two different twists in those filaments. Like you can still tell what's what. And actually, we're still working on this, but like doing this kind of denoising prior to segmentation can be, if you have the right network, like really helpful. we're still working on how to do that properly. Sometimes it just completely ruins your model, but sometimes it's very helpful. But same is true with the denoising as it is with the segmentation, which is just basically like diversify your inputs, like get a lot of things in there. So this is the cytoplasm that I was talking about. This is the same chunk of a tomogram denoised with only small proteins, only large proteins, fake cytoplasm, and then with membranes. And you can see the emergence of the signal in here getting better across here, but it does really sort of plateau here. What does it look like in practice again? So here is a slice through an axon. And these are bundles of cophylactin that form under stress, called cophylactin rods. They fill the cytoplasm in the axon or the axoplasm. And they actually drive all kinds of transport, dysregulation inside the neuron. But here's a single slice through that tomogram after it's been run through our denoising. Yeah, so it's denoised. Microtubules. Bacteria, right? So it's pretty general. You need to be careful though, denoising does bias your data. And in this case, what I've done here is I have a little subtomogram, a synthetic subtomogram of a chromatin array. So these are actually 6 nucleosomes connected by DNA linkers. I denoised them all with a network that was trained to denoise at 5 microns defocus. And what happens is when you run these structural similarity metrics that compare this output to the ground truth, you see that those linkers actually get really dark, suggesting that it's not really a good match between this and ground truth. Whereas when you hit the defocus you were training at, you get this pretty good match. And so again, train at multiple defocus values, and you see that sort of pattern emerge where so one solution is to train a network that has seen all things. Another solution might be to train a network to see things that are CTF corrected, and then you just correct everything before you send it into your network. But I like this one. Anyways, be careful. That went a lot faster than I thought it would. I apologize.
That's fine.
I could certainly just find other slides on my computer, or we can talk about whatever you want. We have 4 more hours together, right?
Yes, we do.
I'm looking forward to it. And we have Dragonfly, and we're going to do some of this stuff. So it's not like you're not going to get them.
Do you have the denoising algorithm you've released, or?
So,
Yes.
Okay.
but only on the bioarchive. And I didn't release the actual model, just how to build it.
Okay, so you still suggest building our own model?
No, I'll give it to you.
Oh, okay.
You're happy to have it. I want people to be able to.
Yeah.
It's very straightforward. And if you have, a certain kind of mixture of complexes that aren't in my model, then I want you to be able to add whatever you want to it, right? And so I like both options, but our next paper is a denoising paper, and it will have some model associated. But in the meantime, just email me and I'll send you 10 different models. Yes.
I was wondering the denoise one, is it just denoising that increases the contrast like that, or are you doing anything special to make the contrast so much better?
Okay. I did my best to optimize, on the screen when I took those shots, I did my best to optimize just contrast for both.
Yeah.
But they were not normalized in any
Okay.
special. But no, nothing more than I don't think. I don't actually know what's going on inside of this network.
So that's, yeah, I feel like that's.
There's a lot of implicit things that are happening, so it is filling in the missing wedge, it is CTF correcting, and it's covering over from some other sorts of harm. In addition to like
Yeah.
the normal like gaps and IP, doing a lot of weird black box stuff.
Okay.
That's the better answer, yeah.
Like, we have not thoroughly tested making denoising models that approach only one of those things at a time or exclude some of them, and they definitely do have different needs. Understand how the network is actually doing those things, see that they are.
How does the denoise interact with your segmentation?
Yeah. I was trying to say that earlier. It can be beautiful. Like sometimes denoise, it's like sometimes I have tried training a model that has both noisy data and like idealized sort of non-noisy data as well. So the network can sort of segment noisy and denoised data. And under those circumstances, it works pretty reliably. But if you take a network that was trained only on noisy data and then denoise with our networks and run it on it, half the time it like really makes things better, and half the time it like tanks performance.
What have you been doing generally? So,
We've been just segmenting noisy data.
what about like noise?
So it's something we're working on for the next, or we're going to work on. I don't have any answers for you. My guess is that it helps. I mean, it's a denoising in general. So the more signal, the better. Let me put it this way, like on some level, the more signal, the better. Obviously that makes sense, but
I've definitely seen this acts a lot of times, just like a fast filter. So I'm like, with your question, that would affect the noise model. Would you be able to maybe manually the noise model for your making your synthetic tomograms?
I don't know if I'm smart enough to do that, but like, if you tell me how to do it, can you mean just like put in a block
Yeah,
of
it's like-
noise?
In like when Matt was doing his act stuff, so we just gave him he extracted what's off that was based on that.
I see.
Like make that put back in.
I don't see why we couldn't do that, but I don't understand the process, right?
I like what you guys said to do, because I had an image processing professor that would yell at us for adding Gaussian noise to tomograms. He's like, the noise isn't Gaussian, don't do it. So, yeah, I like what you guys do, but.
Yeah, we don't really have a noise model in that way, so. By the noise model, we want to track, but like we could, the simulation here is fine, you could make your own noise model that way. And synergy would probably be good, like the very lazy tests that I've done. There are various synergistic advantages. We did reconstructions with like noise assisted program, like our demo or prior care, and then also do denoise secondary regression. Because a thing about regression is it can be extremely aggressive. If you want to recover some sort of signal, you probably can do it. Just may cost at the cost of destroying everything else. You can definitely go.
So I can give one use case, like a colleague of mine, you know, who you may know, I shouldn't say who it is. I don't know if this data is public. They were using our networks and told us that in like thick bacterial samples, like our networks definitely outperformed others in terms of generating denoised cytoplasm, that they could pick ribosomes and other things from that were not working with other pipelines. So in really dense, like especially bacterial kind of situations, it seems to be like working, but it's like better than others. But for the segmentation, I'm still working on that.
How long does it take to train?
We can train some this afternoon. It's like I can train some of these models. It depends. Like if you go and make it like really generalized and you have like 20 different synthetic tomograms that are being input. But I'll show you like what you can do from one 400 by 400 by 50 voxel input. You can make a network that is like 90% of what you need it to be with that amount of data. And it takes 30 minutes to train. So that's why I like to tell people to make your own models, because it's like running a Western blot now. You can just like kind of, especially if you like, we're not going to go explicitly over using CTS today, but like I have tutorials for that as well I can share with people. And you can make these synthetic tomograms in under 10 minutes. So it's pretty fast turnaround if you want to like play around with it. Yes.
All right, I'm gonna, if other people want to jump in. It looked like at one point, maybe you had like a patch-based training and another point there were whole slices. So what do you usually have people use at this point?
Everything is patch-based.
Okay.
In Dragonfly, we use these 2.5D patches. So it's like you pick the outer dimensions and then it's like some number of slices. I always go 11 because that's like the highest I can go.
Okay.
But yeah, all patch-based. So far, I mean, I think all of this would be improved by 3D networks, but currently in Dragonfly, the way that they implement 3D training, and I've done a lot of, it's just time consuming is all I mean. And for the trade-off isn't worth it at this point for us.